it's flexible, so we can adjust it.
Okay, okay. Yeah, the flexible timing is something that is unique to the West, but not so much in our country.
Yeah, here instead of just closing the labs, they gave some slots for everyone, so they can they made a bubble, so we can go in the bubble, 2-3 people, and book our lab for 2 to 3 hours, maximum 4 hours a day. So because in a lab we have more than 10 researchers, so everybody need time to work on their experiment. So we have allowed to go 2 or 3 in single bubble and book for every day. So it can be beneficial for contact tracing. If somebody got the positive, COVID positive, then track and trace is very active here.
Okay, okay. Because I think your lab, no, and that makes better use of your resources also. No, your lab is working down the clock.
Yeah. Yeah, so we work on biological things and many others. So sometimes we can't close it. Like some people are working on cell culture, tissue culture. So they have to go every day and check their cells and freeze, defreeze. So like a material science or physics lab, we can close, or chemistry lab.
Okay.
In biological,
these are difficult.
Yeah, difficult. Even in Christmas holiday, the labs will be open, especially bio labs only.
And there, keeping it open in Christmas holiday means a big thing.
Yeah.
that is their Diwali, you know? Major festival.
They already have lightning in the house, outside the house. They are.
Achha, already?
Yeah.
Oh, okay.
It's almost a month. We know just one day, but here they celebrate for whole month.
Oh, okay. Nice, nice. I think we have another 5 minutes before we can start the talk. I will just see whether my Vice Chancellor also is logged in. Yeah? Just a minute.
I can see him. Yeah.
I'm not... Dr. Mudgal sir login किए हैं क्या? Oh yes, Dr. Mudgal is logged in. Yes, yes. Good evening, Dr. Mudgal sir. Sir, आप muted हैं. आप muted हैं, sir.
That this right was available with you earlier. I was not having this privilege earlier. Now it is made available to me. Anji, good afternoon. And yeah, good afternoon, everybody. And good afternoon, Dr. Nana Sahib.
Thank you very much, sir.
Nice to have you on the platform. And it is so nice of you for agreeing to really take this session to a very, very enriching experience that way. Oh, good.
I think you visited India about what, 3-4 months back, Dr. Thorat?
No, I visited,
before the lockdown.
Yeah, before the lockdown in December and January.
Okay, about a year back. Yeah.
Almost a year back.
Yeah, I have been in DYPU with some guests from Ireland and Poland.
Yeah, Dr. Joanna and
we are there. Yeah.
Correct.
Joanna, she still miss you and your gesture. And
it was a lovely personality.
Yeah.
I remember meeting her and very warm. And
unfortunately, she had got infected with the COVID, and I think she is on leave for long.
Oh, okay.
Yeah.
Do convey my wishes to her. Actually, if you can send me,
of course.
In fact, I will send her my wishes personally also. This COVID, what is the COVID like now in UK?
UK has, every day they have some cases, more than 10,000 something, but they already did a lockdown in November to control the spread in December. In Christmas time, people are moving here and there, so they are visiting their families. So they made a very good trick actually. So they controlled before the Christmas. And in the Christmas week, there will be no lockdown, so people can visit to their families and friends only, not
travel.
Yeah,
that would be useful.
Still, there's a widespread of this, right? Still, it is spreading there in UK.
Yeah, it's actually old people, they are very much
Because
infected.
comorbid, that's one issue.
Yeah.
And some people are what to call speaking about that people who have deficiency of vitamin D, they are highly prone.
Yes. Yeah, here
Especially
it is a major issue.
In a country like UK and US and some portion of, definitely there's a
These
lot of population.
northern countries, they don't get good sunlight, so they
Sunlight,
didn't
yeah.
generate. Yeah.
I don't know how far it is scientifically proved, but this has been valid.
This is vitamin C, vitamin D is necessary to, it is a boosting for our immunity.
Because there was some study they were talking about from University of Washington or something that 80% people who were suffering from COVID are the deficient into vitamin D as well.
Actually, vitamin D deficiency has been associated with so many infections, right from tuberculosis to COVID, that creating a positive kind of relationship is
here in UK or in Europe and the USA, the another scenario is that in the winter they get a flu every year. So this is combined effect. Flu plus COVID. So in flu they have every winter the mortality increases by 40%, and now this year it is a COVID. So that's why the mortality is high here in winter.
And
Especially older people.
10,000 cases for a country the size of UK is pretty big.
Yeah, pretty big.
I mean, a country India's size is dealing with 20,000 odd cases every day new. So 10,000 is a big number.
Big number.
Anyways, sir, with your permission, should we start?
Yeah, please, ma'am. Yes, please. Yeah.
So, well, I can see Dr. Lokhande has signed in, but anyway, very warm welcome to all the participants who have signed in. Some have signed in very promptly, so thank you so much for being so very well on time. That way we make maximum use of Dr. Thorat's time with us. Before we go ahead, I would request our Vice Chancellor, Dr. Rakesh K. Mudgal, to say a few words to welcome Dr. Thorat, after which I will give a short introduction of Dr. Thorat, and then we'll hand the stage over to Dr. Nana Sahib Thorat. Dr. Mudgal, sir, please.
Thank you, ma'am. Thank you so much. And it is so great feeling welcoming our own alumnus, Dr. Nana Sahib Thorat, for this session. You have a very, highly respectable name in the community within the university and nearby, and we are all proud of you. Because having our own student addressing us, it is really a great feeling, and we are all fortunate for this particular session, and definitely we look forward for further more sessions of similar nature with the people who have contributed immensely. And let me share with the participants, Dr. Nana Sahib Thorat has brought great laurel to this institution, and whatever possible, scholarships and recognition as a young researcher, he has been blessed with all such titles and accolades from every corner, best of the institution in the country, and we wish him all the best. That is what, as we all know, that research is becoming more and more important, and especially for a country like us, which people are putting into a bracket which is not so highly recognized as far as a research output and contribution to the body of knowledge is concerned. But with people like Dr. Nana Sahib and of similar caliber, definitely this particular thing is going to be a story of past with the dedicated effort. Not taking much time, it is a fortunate moment for us to really listen to him, because he has done a pioneering work in the field he will be talking about under these testing times. As far as university is concerned, definitely it is being put into a great focus into the institution community to bring research and consulting activity on the front floor. And for that, every activity will be undertaken with full dedication and determination to take it to a concluding end, and then further taking it to the higher trajectory that way. But when it comes to research, definitely it has got a direct connection with the creation of knowledge or reproduction of knowledge or dissemination of knowledge. And for that, the essential thing, which I normally speak it out, that it is a way of thinking, how we are able to think, and which has got two essential components for all of us to appreciate. The one is the critical thinking, and another the creative thinking, because unless we are able to critically examine any issue, we will not be able to identify the right problem. And once we identify the problem, definitely creativity is needed, because what are the various approaches to solve that particular problem and benefit out of it is definitely are going to the fruit of critical and creative thinking that way. With these words, I request Dr. Shimpa Sharma to take this particular session further, because everybody is so anxious and enthusiastic to listen to Dr. Nana sir.
Thank you, Dr. Mudgal, for those few words. Before I hand you over to Dr. Thorat, I'll do a quick introduction. As Dr. Mudgal said, it's an honor to be able to introduce someone who was a student in this university at some time and has gone out in the world to achieve such great laurels. Dr. Thorat is an outstanding and a very experienced researcher who's currently working as a Marie Curie Fellow research scientist in the Medical Science Division of the University of Oxford, United Kingdom. He is a two-time winner of this most prestigious fellowship, the Marie Curie Fellowship. He also now works as an evaluation panel member of this Marie Curie application for the applications which come in the field of life sciences and engineering sciences since last year. He has published 50 peer-reviewed journal research articles and papers. He has one international, one European patent filed, 2 European innovations or inventions registered. He has written 5 books and 10 book chapters, and all, I don't think he has yet hit the 4th or the 5th decade. A most sought after speaker, he has presented 5 keynote speeches at conferences, 20 invited lectures, and I'm sure the number is just going up every week. 5 oral presentations at prestigious scientific peer conferences across the world. He has been represented in Singapore, France, Germany, USA, Poland, Ireland, South Africa, India, and I think the list goes on, South Korea. He has generated research funds in excess of 6.5 crores, and he has supervised students, junior researchers, extremely active in outreach and scientific dissemination in the service of the wider community. And this talk is just one more step in that direction. He has received international as well as national awards and acclaims for great amount of research contributions. And the list is long, but I'm just going to mention 2. One is he has received the fellowship from the Japanese Society for the Promotion of Science in 2017, and the PBC Outstanding Fellowship from Israel in 2016. He also has experience of having worked at the Samsung Biomedical Research Institute in South Africa for a period of almost over a year. He is right now working in the Medical Science Division of the University of Oxford, jointly with the Engineering Science Division, and his current research work involves designing new light-activated nanomedicine therapy and the Raman imaging techniques for brain cancer of pediatric patients. Earlier this year, Dr. Thorat once again made India extremely proud by becoming the first Indian awardee of the European Commission's Innovation Radar, the Grand Prix of the Innovation Radar Prize 2020. Congratulations, Dr. Thorat, on this latest achievement. We are privileged to claim Dr. Thorat as alumni of our university, as an inspiration to a lot of students, both in our university and outside, and we pay our sincere regards to his guide, Dr. Shivaji Pawar, former Vice Chancellor of this university, for playing a major part in his growth. I thank Dr. Thorat for accepting our invitation for this talk, and especially for addressing such an important topic as the COVID vaccine. Dr. Thorat, may I please invite you to take the stage, and the mic is yours. Thank you so much.
Thank you very much, ma'am, for nice introduction. And I would like to first thank Vice Chancellor, Professor Rakesh Mudgal sir, and Chancellor and the President of our university, Dr. Sanjay Patil Sahib, Registrar Bhosle sir, and Dean for Research, Lokande sir. And some of my colleagues are also attending, so I'm happy with them as well. Thank you very much for inviting me. And though I am not a medical practitioner or the clinician, and even I'm not working on the directly or involved in the vaccine development, but now we are starting some new liposomal activities for vaccine delivery. And our lab just a week ago decided to start this project. And me and another colleague in my lab who will be heading this. But my prime work is more on cancer, and I'm happy with that, because from last 10 years, I'm working on this field. And not a specialist oncologist or something, but we are just heading towards the future of the technology. And I'm sure after 5 or 10 years, we'll see some new developments in oncologies, and some technologies that what we are developing here or worldwide, they will have good future in oncology, in therapy, diagnosis, and even in therapy monitoring. So this is all about my what we are doing. So right now I'm working in medical science division. It is a big, it has more than 21 departments in medical science division in Oxford. My affiliation is with the women's reproductive health. Actually, it is very different, and I never seen such a department in any medical, but here they have very fancy departments in medical divisions as well. But my work is more on the engineering side. We take the chemical engineering department as well as our commenter. The Oxford is very well known university across the globe. It is one of the oldest university in English speaking country, situated in the core of the UK or England, and very far, not far from London, just 1 hour from London. Oxford is a small city, and it's just a student city. There are two universities. One is Oxford University, and one more is the Oxford Brook University. Here, universities are not campus based like us. So in many Indian universities, they have their own campuses outside the cities or somewhere in city as well, but they have their own campuses. But in Europe or UK, you will find universities across the cities. So Oxford is also like that. So this is whole city and university. So we can't differentiate where is the shopping markets, where are residential complex, residential building, or university building. So it is very tricky here to find the departments and a shopping center. So all universities are actually across the cities. Oxford is the same. The difference between other universities and Oxford and Cambridge is that they have colleges as well. So like the Trinity College, Magdalen College, these are very famous colleges where many Nobel laureates and prime minister were educated. But this is a kind of whole university buildings, very old buildings, 500, 600 years ago they built it, and still they maintain it. We here, you will be surprised, we can't take any photo as well here or the video shooting. We have to take a permission from here local council, because this is World Heritage and it's not allowed. So many times you have to take even especially professional media, if they want to shoot something, they have to take permissions for every building. Oxford is famous because of their curriculum, their objectives towards research, faculties, and overall impression by many Nobel alumni. Like Oxford produced 40 countries' prime minister. In India, Manmohan Singh and Indira Gandhi, they were educated from Oxford. More than 80 Nobel laureates are from Oxford, or they are jointly with Oxford. And right now, we have around 40 Nobel laureates. They are teaching in many departments and doing research. Even our department, medical science division or some chemical division, we have 2 Nobel laureates alongside. And they have maybe European, UK, or US patent or international patents. And they have huge investment for technology transfer. So even we are working with the technology transfer division. So just not doing the research in lab, but we are alongside working with the professional who has experience how to transfer our daily lab work into the technology and market. So we have especially confusion or dilemma. What is the difference between just taking IPR right and technology transfer? So when the most of the laboratory work, first they all scientists they try to publish in some research journals, and this is kind of basic science. Even they are developing technology, but publishing in journal, it is, let's say, is just a science that what we are exposing to the society or the scientific field. Then it comes to the patents, where we are taking our own rights for the what we did, but commercialization is the next part. Even we have so many patents. Patents are just a right for our own activity, but commercialization means finding the right market and what is the current status of our technology, our products or technology, and then making a venture or our own companies. So just like Simpa ma'am told about my recent award, Innovation Radar prize. So that was the one of the best example how we translated our lab research into the technology. So last 2 years when I worked in Poland and Switzerland, we on the first day we decided we will not publish anything. We will not, just because we have already around 50 publication on my name, and just we are publishing 2-3 more. It's just counting numbers, not something doing different. So we decided to transfer, make the technology. And even in Poland, we have technology office there. And from the first day, we started to work with them. We first applied for patent. There are many ways. Even we don't have data, we can apply for patent or we can file patent. So first we filed patent, we took IPR right. Then with the help of technology transfer office and IPR office, we designed the innovation and invention of our product and our technology. And fortunately, Europe has many opportunities to showcase such invention to the people. And we got selected for such invention prize. And fortunately, we got it. And then now we are trying to establish our own company somewhere in Europe, because this award gives us the freedom to establish our own firm on technology base or product base or even consultancy base. So this is kind of a research culture here. Even India, we are little bit lagging in such applications, and that's why many of technologies are coming from outside. So this is all about the brief introduction, what we are doing and where I am right now. My research, actually, though I am in Oxford, my research is totally funded by the European Commission. European Commission is a bureaucratic body of European Union. European Union is a body of 28 countries. Now UK is leaving the European Union from next month, but they will be involved for research activities. So European Union is funding a huge amount for the research from 2021 to 2027. They already have 7-year plan, and that plan has around 100 billion pound. Means it is more than 1 lakh crore rupees just for research. And fortunately, just two days ago, I have been selected by European Commission for their beating cancer plan. So they have 7-year cancer plan. And they have selected 17 planning and commission members, management members across the Europe. And I already have been involved in many European activities. So they again selected me for next 7 years. So I will be involved in not only just research, but policy making as well. So what will be the future? What new direction should be funded? What new direction should be focused for research? I will just give the brief a single slide about what we are doing in the infectious diseases, because today's talk is more on COVID and its consequences. So you all are, real practitioners than us, because we are just a researcher and we are just reading and then implementing our ideas. But you are every day exposing and practicing the ideas. So infectious diseases are two kinds. So they are from viral or bacterial. There are some others like malaria. It is not categorized in bacterial or viral, but they are in different kind. But we work on some infectious disease that is called the sepsis, and it is very dangerous overall worldwide, not only in any specific country. So sepsis is just it's due to the drug resistance bacteria. So we recently designed a technology, laser-based technology and nano technology. So we designed gold nano capsules. We call it gold nano capsules. So it has a gold nano rods, very small, around 30, 40 nanometers, and they are conjugated with the biomolecules like aptamer. Even we use the Ayurvedic product curcumin, very pure product of our turmeric. And we designed the antibacterial therapy. It's just not antibacterial, but it is controlling the biofilms in static condition, in biofilm conditions. It has a wide absorption of light from normal visible light to infrared light. And that infrared light absorption gives us the photothermal therapy effect. So we designed the photothermal instrument and technology in our lab, and we applied it to kill the bacteria in biofilm condition and static condition. And these are not normal bacteria, but it is a clinical and community spread strain, USA 300. It is actually a very dangerous and just has a not long history. It is evolved from 1995 somewhere in US. And now every hospital across the US and Europe is infected with such a strain because it is a drug resistance. We call it is a superbug. So we worked on superbug, and unfortunately, even in India, we are not allowed to work because we need some biosafety laboratory protocols, and very few laboratories are there, like BSL-1 to BSL-4. So we need a BSL-3 or 2 laboratory. So we had to set up in Switzerland and Poland where we worked on that. And this technology we patented and we published as well. So this will be a journal cover page of one of the Royal Society journal. So here is just our work, where we are working in infectious, but alongside we are working more on cancer side, and my prime focus is cancer. So how I came in vaccine and these all information. Actually, it is just worldwide you will find two type of researcher or scientist. One, they do the very hard work in the labs, but they can't explore it to the community because they don't understand how to communicate. They know how to design the experiments, publish it, and it is I would say 90% of scientists in this category. They do very hard work, but they don't explore it. 10% scientists, I can't say they don't do hard work, but they have the extra ability that they can spread the knowledge to the society. And from last 6 years, when I involved with the European Commission insisted us to just don't do the research in the lab, but try to spread the knowledge to the society. And I get trained with from different scientists across the Europe, how to transfer this knowledge. And when we understand some scientific terms, just try to explore to the society. I remember in 2015, from starting 2015, I worked with the small school children, and we started to one program like Science for Children. We designed very small instruments, scientific instrument. We are exploring it to the school children, how science works, what they are learning in the school, how actually we do research on the same. And then I realized that, okay, even I don't have background with the medical field or the clinics. I can explore the knowledge. I can understand the English and scientific terms behind the vaccine development, behind the coronavirus. It just, we need to explore to the society. So from last 6 months, actually, I involved with the media, print media initially. Then I came to in contact with the TV media as well. I got interviewed by TV media here in Europe and in India as well. And just I'm exploring the knowledge, what we are getting here in Oxford or through the knowledge partners to just a society. So this is overall about me, how I started my COVID research or knowledge transfer. So become a physicist, I would say. I took a chemistry as a bridge to transfer my knowledge in biology. And as interdisciplinary researcher, I work on different fields. So still I'm using physics, chemistry, biology, and exploring in medicine. So I have just a normal score in all my 10th and even in my all curriculum. So just I'm explaining why it is because I have my own degree in physics, but now I forget about everything about physics, and just I'm every day exposed with this knowledge of oncology, knowledge from infectious diseases, or the research work from molecular biology to the protein chemistry as well. So in Oxford, what we are doing in Oxford, actually, we are working on brain cancer and designing a device and doing animal studies as well. Especially in brain cancer, we focused on pediatric brain cancer, medulloblastoma and neuroblastoma. So they are medulloblastoma is really a threat to the pediatric patient and not a good cure for them. We can't treat them with the chemotherapy or radiotherapy because they have many side effects. So we need some new technology and new diagnosis and therapy monitoring as well, because they have very small children and we don't know what time they will replace again. In my 7 year of span after PhD from DY Patil University, I had exposed with many different fields and different ideas, different people, and many scientists across the globe. During that 7 year, I developed my own career in interdisciplinary things. So from publishing books to patenting ideas, applying for funding, getting own funding, and publishing papers, and all outreach activities as well. So we have scientific collaborators across the globe and top universities. I'm surprised. I am fortunate that I have one collaborator from University of Zurich. You will be surprised. It is one of the top hospital, animal hospital, where we did the MRI for horses and dogs as well. So they are very sophisticated hospital in the globe. And even I'm fortunate, still I am working with my colleagues in DOIPU and Kolhapur. So with this brief introduction, I will come to the most today's talk and the today's part, that is the COVID vaccines and what is stage. And though you are all expert in the clinical side, there are some basic things that we need to explore and we need to know. So let's start with what is the current stage of vaccines and how many vaccines are in pipeline and what type of vaccines are there. So right now we are more familiar with the Oxford AstraZeneca. In India, it is Oxford Serum vaccine, Pfizer vaccine, and Moderna vaccines, because they initially took the risk production and research before knowing the real threat of the pandemic and will it be implemented well. But they took a risk because they have big pharma giants. But later on, many other pharma companies invested their part in the vaccines. Most of the pharma companies, they don't like to invest in the vaccines. So we all know why it is, because vaccines are generally developed for the viral and infectious diseases, and their lifetime is short. So we have to invest a lot for short time, and in return they get very small benefit. So I have been involved in one project with Pfizer and Janssen in 2017, and I know how they work. The pharma companies, how they really work. So they always find the money. When we have some ideas and want to explore with them, they ask first thing, can we generate the money from this? So they are very straightforward because they want money. But vaccines, they gives them money, but it's not that what they get it from the drugs like for cancer or diabetics or cardiovascular. They say it is a green evergreen diseases for them. Right now, more than 40 vaccines companies developed or clinical 2 phase, but only 9 were in clinical 3 phase, and Moderna, Pfizer, and Oxford, and Sputnik, that is Russian, they actually had a clinical 3-stage data in their hand. Others have just still doing the phase 3 trials. They have different technologies as well. I will come to that, all technologies there in short. What mechanism of and how they are different from each other. So what is the current status in vaccines? So many vaccines, we need 2 doses. Johnson has recently announced that their vaccine will be single dose. I will come to that as well, why we need 2 doses. But some vaccines we need 3 doses as well. But still we don't know which are they, and their clinical 3-phase data is not published, or they not announced. So we don't know which vaccine we need 3 times or 2 times. Sometime here in UK, many researchers thinking that we don't have to be rely on single vaccine. We need multiple vaccines to get there, because we are more than 8 billion, 7.5 billion population, and the production companies production limitation, like Pfizer can produce only 1 billion doses a year. Oxford Genica can be around 3 billion doses a year. So there are production limit for everyone, and this developed country already secured all doses, like USA secured around 1 billion doses from all companies. India around 1 billion, European Union, whole 28 countries around 1 billion. Unfortunately, African countries, they did not secure some. They have few millions only. So it's a real challenge for them as well. Many developing countries, undeveloped countries have threat of this disease in future. So we need to rely on different vaccines as well. And scientifically as well, some combining 2 vaccines can be boosting the immunity. Here we can understand why we need 2 doses. So all the data from these 4 companies, from Moderna to the Sputnik, the initial dose, they trigger their primary immune response, but with the time, this immune response is declining, and at a certain time, we need the second dose, so we can be then boost the primary response to the secondary. So secondary response is higher and giving the more immunity, and it is generating the memory to the immune cells. And that's saturating over the time. So we need the memory as well, not just producing antibodies. We need the memory because in future, antibodies lifetime is very, it's just for a few days or let's say some few months. We need the immune memory. So in future, when the real virus comes, so memory can trigger the antibodies again. And that's why we need 2 doses. Many companies, even Oxford, they are the top university and top medical school. They made a conflict in their own data as well. And they are now doing the stage 3 trials again in USA because of the different doses. The story is very simple, that the techniques they used to monitor the inactivated virus particle in the vials in Oxford is different than the company produced in Italy. So Italy, they produced all vaccines in Italy. They manufactured it in Italy. And the technology used to count the inactivated virus particle in the single dose was different there. So that was the big problem. And now they are doing trials again. So this happens in when these vaccines were developed in very short time. Worldwide, I would say Europe and the USA has dominated in pharma industries. And that's why they secured all doses initially. Even Europe is the first. When before even people were thinking about the vaccine, they secured around 1 billion doses in May and June from Pfizer, Moderna, AstraZeneca. They will not take the Russian vaccine because of the geopolitics, but they secured all these vaccine doses. And then India came, then Australia. Australia has their own pharma companies, and recently there was a big mess with their invention as well. I will come to that at the end of our talk. This is current scenario of vaccine and their trials. So trials are ongoing in India, Europe, South Africa. Very few African countries are involved. Brazil is there now, Argentina, US, Canada, Russia, Japan, Australia, Vietnam, Myanmar are now involved. China is developing some vaccine, and they are taking trials in Pakistan, but it's not officially, they did not announce it. So let's come to the very brief about the vaccine. So you all know about that. I will just give the introduction about this, how vaccine work and what are the new technologies coming. So vaccines are three types. They are 3 to 4 types: DNA vaccines, RNA vaccines, and protein vaccines. DNA vaccines are well known, and already we are using that. RNA vaccines are new, like Pfizer and Moderna. They are all rely on the RNA vaccines. Some are protein vaccines there, but still it's on research level. RNA and DNA, I would say RNA vaccine is the future of vaccinology, because now technology, we can manufacture RNAs in huge amount. Earlier it was not. That's why we rely on the DNA vaccines. How DNA vaccine works, actually. DNA vaccines are very simple. So we take some strands, DNA strands from that antigen, and we use the plasmids. And especially people are using the spike protein from corona, and there's a small part using the genetic engineering. And they are combining it with the plasmid and transferring it to the cells, and then cells transfer into RNA, and then start to produce the proteins. So simple technique in DNAs. In RNA, actually, this is a new technology. And here we are now starting our work, designing a new liposomes, these lipid nanoparticles. All those Pfizer and Moderna, they use these RNA vaccines. And why they use RNA vaccines? Because the scientists behind these two companies, they are actually the nanotechnology scientists. One group from Germany, they are actually Turkish scientists who were designing the immunological they were immunologists and using the nanoparticles for immunology, immunotherapy. And they got a breakthrough with the mRNA with different vaccines, but then they diverted their work into the coronavirus vaccine, and they designed the mRNA, and they use these lipid nanoparticles to conjugate the mRNA, because we know RNA is not stable in the blood, it degrades, and blood enzymes, they attack on RNA, and we don't get a real effect from this. So that's why we need some carrier to deliver into the right place. And this is first kind, like even Moderna scientists, they are from MIT, USA. And these are huge giants. They are big scientists who developed many lipid nanoparticles for cancer drug or other drugs delivery. And first time they are trying to use with the vaccine. This Moderna company is very new. It's just established in 2010, and it is a kind of spin-off. And this is their first breakthrough. And they got it. They are now billionaire. Just they started with some million, but now they all, their management members, they are now billionaire because of just one invention. RNA, DNA vaccines are future, but there are some other vaccines as well. Like vector vaccines, we call the Oxford. They developed the vector vaccine. So this is a new technology again. First time it will be implemented, but they already developed for SARS and MERS and Ebola as well. So Oxford, they have their unit, vaccine department. It is established in 1916, and from 2000 they started to find the vaccines and drugs for disease X. So they use the modeling and artificial intelligence technique to find out the disease X in future. And before knowing the disease, they started to work on the vaccine and drug discoveries. And this is a breakthrough from that project. So they were discovered the vaccines for SARS, MERS, and Ebola, and they used the MERS vaccine technology for the coronavirus. So what they did the live attenuated vaccine. In that they took a viral vector of adenovirus. It is a virus for the chimpanzee monkey. It is causing the flu to chimpanzee monkey, and they inactivated it, so it can't be get any threat to the human body. And then they removed some part from that virus and joined the S protein part to that vector. And then that adenovirus is used as a delivery platform to S protein. So it can be go into the cells, and then they will trigger the immunity. So these vector vaccines are really very simple to design, and we can do the mass production, like even with just the simple technology transfer. Serum Institute in India already produced around billion doses. They are ready for they are just waiting to get a permission from Indian government, UK government, and European Union, because these all be coming here as well. So already how many doses are in, manufactured. It's very simple, but they have, very efficient, not highly efficient like Pfizer and Moderna. So why Pfizer and Moderna vaccines are efficient? So let's get a very simple example. If we want to travel from Kolhapur to Mumbai, we have many options. Like we can go by the bus, we can go by walk as well, but we can take the helicopter as well. We can get the flight as well. But helicopter flights, they are expensive, but they are very efficient. So Pfizer and Moderna vaccines are like helicopters. They have new technology. That's why they are efficient. So helicopters like liposomes. Liposomes manufacturing is very expensive. Here, just if you want to buy one surfactant for the liposome, it is just 1 milligram cost around $300. So it's very expensive, but it is efficient as well. While the Oxford vaccine, that depends on the vector and some new different technology, it is kind of a bus where we can accommodate many people, but it's not efficient. We have to take around 6 to 8 hours to go Mumbai, and we can't do many multitasking simultaneously. That's why when the Oxford announced their results, they have only efficiency around 70 to 90%. Let's say 70% is optimum, but the Moderna or the Pfizer, they have 95% efficiency. But on the other side, they are expensive as well. And we need a deep freezing and huge investment in infrastructure if we want to vaccinate everyone with the Pfizer and Moderna. So this is, pros and cons for every vaccine and even for every drug as well. There are some other vaccines, live attenuated vaccines, that already been used for some diseases, but recently it is implemented by some universities in UK, and one Indian company is designing this type of vaccine. But I will come to this vaccine, inactivated vaccine, that all we know. We already have these vaccines for polio and other diseases where we are inactivating real virus, which cannot make a threat to our immunity or our organs, but it can generate the immune response. But it has again have some cons with these vaccines as well, because it has have short lifetime there in the cells, and their immune response sometimes they don't get the immune memory response as well. But in India, actually, this was the very successful project. I'll not go with this in detail, but how this mRNA induced the antibodies. So this is just a simple biochemistry. Probably you have this the knowledge, where this messenger RNA trigger our memory cells to produce the immune cells to produce the antibodies, and even they generate the memory to the be anti cells. So we can get same antibodies in future. What are now current risk and responses to each vaccines? So like DNA vaccines, nucleic acid vaccines. So their integration mutation is the risk because virus is mutating. Like SARS is mutated in MERS, MERS is mutated in COVID. And there we need a very special facility to manufacture it. Some RNA vaccines actually safe, but still there is a big debate on the mRNA vaccine because mRNA is a genetic material and they took it from the virus actually. So people, even 50%, they know about biochemistry and vaccines, how they produce or how they trigger our immunity. Many scientists and even common people in Europe and UK and USA, they feel that these vaccines are dangerous because if they interact with our genetic material, probably it will cause some carcinogenous effect in future. And that was the big threat with mRNA vaccines. And that's why it was not implemented earlier. But now Pfizer and Moderna took risk with these mRNA vaccines. Every vaccine have their pros and cons in terms of production, manufacturing, or their infrastructure, carrying to different countries. Not all countries have facility to manufacture these vaccines. Like Pfizer have their single production facility in Belgium, and all vaccine now coming in UK, it is from Belgium. Like Moderna, even they don't have their own manufacturing unit because it is very new company. So they are now finding manufacturing companies in Australia, Europe, or USA. Unfortunately, in India, I'm not sure that Moderna vaccine will be there because we don't have such a technology. Even we don't have such a specialist there. So don't know about how these lipid or liposomes are used to deliver these vaccines. So this is another threat and risk for vaccination in India. This is really a new technology now in the vaccination. So we all know about vaccines are delivered by different ways, from nasal to oral or intravenousal. And right now, nanomedicine, nanotechnologies are developed. We are working on some inorganic materials, but I'm not sure these will be used for vaccine delivery because inorganics have high toxicity than the organic materials. But the liposomes are beneficial and they already have their good data for their biocompatibility and toxicity. And that's why they used it. So I'm almost at the end of my presentation. This is the plan now US and Europe has made how vaccination will be actually in UK vaccination started 2 weeks ago. Pfizer vaccine, many people just yesterday they have announced how many people get vaccinated. So around 1.5 lakh people get vaccinated in UK with Pfizer vaccine, and all they are about 80 years and primary health, frontline health workers, not all health workers, but practitioners and medical doctors who are doing surgeries every day, they get vaccine. My friend here who is working, he's a Marathi guy, even he did MBBS from Mirror. So that guy has doing surgeries every day in Oxford Hospital. So just 3 days ago, he get vaccinated with Pfizer vaccine. So some people who are frontline health workers, they get vaccinated. So the big here, even UK and Europe are now making a plan in big, they are reserving big conference centers and football stadiums for mass vaccination. So they are now doing the infrastructure like check-in in our airport or bank counters. So where people will be coming with social distancing, they will have their some identity card and something from their here in UK we have NHS, National Health Service. So they are giving some identity for everyone or some number. So they will have their card or something, so they can be called for the vaccination. They will be giving vaccine in some rooms individually. They already trained more than 1 lakh nurses and health practitioners. So they given a training to them, and they started it from June. So they prepared very early for that. Our threat is we have to train, those nurses or doctors in rural areas how to vaccinate, and there may be some initial side effects after the vaccine, and we need to know. First, they have to know, then they can be explored to the common people. So they will have again some rooms where if somebody has a immediate reaction with vaccines, some allergic people get a immediate reaction with the vaccine, because vaccines have they have actually foreign bodies to our organs. So there's immediately our immunity immediately oppose that initially. And so there are some allergic reactions as well. Some people in UK, old people, they get this reaction. And overall, they will have this kind of, infrastructure. So people are coming with this way and they will be going on the other way. So with maintaining social and other distancing. So this is a plan that they made, and they just yesterday they have outlined their plan. Even India has made a plan. They documented it. So we have to implement such things in India as well. So now I will come to the final, that what is the current stage of Oxford vaccine and when it is coming. So the Oxford vaccine is the most appropriate for India because it don't need freezing temperature or -70 temperature like other vaccine. And we have already Serum Institute who is producing billion of doses, and they have experience with that. And we have infrastructure for that. We don't need special infrastructure like -70 freezers or carriers for that. Like Pfizer vaccine, they have made a suitcases with a GPS monitoring. So it is very expensive as well. So we don't need such a technology for Oxford or the vaccine developed by Russia. But unfortunately, I would say as a medical practitioner or as a scientist, we have to be open about our data, what we are doing. So Oxford and Pfizer, Oxford recently announced all their trial data and results in Lancet just on 10th of December. And Pfizer, they announced in New England Journal of Medicine just 2 days later, the Oxford. So they are open. Earlier, all these pharma companies, they never announce their results in any publication or in journals, because they always hidden their protocols, how they did stage 1, stage 2, stage 3 trials. But now European Union or US FDA and all governments and people now well educated, and they want to know about vaccines. What are the constituents part of vaccines? What are technologies there? How they are implementing phase 1 to phase trials? And I hope the Russian vaccine, they will announce their technology and how they did their clinical 1 to clinical 3 trials. Because these all vaccines, they developed in very short time. So we need to know about that. Why we need to know about that? I will give you the very simple example. Recently, it happened in India. So one media, Marathi media, they misleaded some scientific information coming from the Australian university, Queensland University, where they developed one vaccine. And they said that this vaccine is causing the HIV positive. So people who get vaccinated, they have HIV virus in their body. But actually, it was not that case. So what was the case? So what was the vaccine developed by Australian scientists? So they used 2 different protein constituents. So one from coronavirus, and by mistaken, or why, I don't understand, why they took the protein part from the HIV virus. Because they believe that protein is delivering or boosting the coronavirus corona protein efficiently. So they are combining corona protein and HIV protein together has a higher effect, and that's why they choose the HIV protein. But unfortunately, when they did it, they know that earlier that when we use the HIV protein, there might be some antibodies will be generated against that protein. So they already informed to the these all people who get a trial for phase 1 and phase 2 that they will get some antibodies for HIV protein. But unfortunately, they believe that 5 to 6% antibodies will be generated against the HIV protein, and all will be against the corona protein. But when they examine after the trials, they found that around 40% antibodies are produced against the HIV protein. So it is a huge number. Actually, their main aim is to make antibodies for corona protein, but we can't predict the lab results or animal results with human. So human body is completely different than the even animals like mouse or others. And then some of media approached me about to explore the technology, what is reality. Then I wrote one late article which were published in all Maharashtra edition, that what is actual situation about that vaccine, and it is not infecting us as HIV, but it is giving some antibodies against HIV, which is not our real aim. Our aim is to make antibodies against the coronavirus. And then Australian government and their scientists understand their mistake. Even though it is a good vaccine, they stopped that project. And this vaccine is not coming to India, and the project was stopped. But the scientists will be working on this vaccine. So they are not stopping the real project, but they are not manufacturing this vaccine at all now. So this is many consequences will be in India as well. When many people, you know very well, all doctors, they know, when you treat any patient and you ask their history, always patients want to hide their medical history. They don't give you the proper information. Are they ill, or they have taking other pills or drugs? They always hide. And you know, vaccination, we don't know their real side effects, their efficacy, long-term side effects. So we need to know the medical history of all people who are getting the vaccinated. And this is a big threat for vaccination in India because people will hide their history and they will get ill because of their history, but they will, tell that it's because of the vaccine. So some people, we know HIV virus, we can't detect within 1 or 2 days. We need at least 6 months. It has an incubation period of 6 months, and then we can detect it. And we can get their symptoms after 1 or 2 years. Some people will, those who have HIV virus in their body, and they will get the corona vaccine. And later in 6 months or 1 year, they will find their symptoms. And then they will say, oh, it's not because it's not my mistake, it's mistake of corona vaccine, and it's because of the vaccine, because news were spread. So that's why Australian government stopped this project. At some time we have to choose the right decision, even though they have invested around $50 million in that project. So at the end, I would say thank you very much for listening, and this is my great privilege to talk with the doctors. I'm not a medical researcher, and as a researcher, we are trained that always don't talk or don't spread the one-way knowledge. It must be two-way knowledge transfer. So now I am open to answer your questions, and I hope you will not answer tricky questions to me. Thank you very much.
Okay, thank you so much, Dr. Thorat. It was wonderful. I think we have some children who are enjoying your talk as well. Before I go to the tricky questions, can I ask you very simple? It's a kind of a personal question. Someone wants to know whether you have taken the vaccine and what was your experience if you have.
No, we did not. Actually, I came here in Oxford in last week of August. That time, all trials were paused, stopped. So probably we will get vaccine. They have the protocol. So right now they are giving vaccine to care home people. So care home people, like we have care home as well. Here government, some people above 80, 90,
Okay.
they can't stay in their own homes, so they go in the government care homes where facilities, there were nurses and health workers. They take care for them. And unfortunately, in UK or in Europe, in even USA, the high mortality is from that care homes. So all they are very old age people, and even they have living in a kind of a hotel where 100, 200 people sharing, rooms, and they have their own separate rooms, but they gather every day, and that's why their infection rate is high. So they are first giving to them, then a frontline health worker, and then above 70 years, 65 years. The young generation will be at the last. Same will be in India, same will be in the USA, everywhere in the world.
I think globally everybody is going to go from high risk to low risk, so that
Yes,
order
yes,
will
yes,
be
yeah.
followed.
Yeah.
Tell me,
Children
but
will be last.
there are some questions our people have got. One question which has come on both the YouTube as well as here is, people who have already been infected with COVID infection, do they need to take the vaccine? And a correlate of this question is, if somebody does take the vaccine, how long does it work? What is the length of efficacy, duration?
Yeah, so in one slide, I show the why we need a first dose and the booster dose. So it's all depend on how our immunity response. So those who first infected with the COVID, I would say if they have the antibodies in their body, we don't need to be vaccinate them first.
Okay.
Yeah. If they have the memory, because the antibodies is different and the generating memory against that virus is another part. So if they have that memory, then they don't need to be vaccinated. But those have small memory. Some people, they get a second time COVID as well. Somewhere in Mumbai, some cases were there. So probably we need to vaccinate them as well. It depends on now how government prioritize. It's not on our hand, or scientists or doctors can't choose now. It's all government policies will be preferred. And they don't need to be worried about that. It's depend all about symptomatic and asymptomatic as well. So vaccines are working on asymptomatic things. I missed the one thing. I will just tell you what difference between Oxford trials and Pfizer and Moderna trial. So Pfizer and Moderna, what they did, they gave the in clinical 3 trials, they gave first and second dose, first dose, and then they asked all the volunteers that come or report us if you found any symptoms about the COVID. If you don't find any symptoms, don't report us, even in the second dose as well. So they did not monitor them, the volunteers, but exactly opposite what Oxford scientists did. They asked, they did every week the swabbing from these volunteers. So every week they monitored after vaccination, these people are getting COVID symptoms or not, or the COVID infection or not. So they found that every week that they were monitoring. So they found there were some asymptomatic patients as well. Even they are vaccinated, they have infected with the virus, but they don't have any symptoms. So this is asymptomatic. So vaccines will be just in right now, many scientists, they are just advising that vaccines is just to stop the hospitalization, a burden on hospitalization. Everywhere hospitalization, the health system is collapsed. So vaccine will stop that. And in latter, it will generate the herd immunity.
It appears this vaccine is going to work in reverse like the dengue vaccine. In the dengue vaccine, they said those who have been infected only should get the vaccine.
Yeah, it's exactly
this
opposite.
This is working the other way. Take the vaccine so that if you get infected, you don't get a serious infection.
Yeah, just I read yesterday about dengue vaccine. It is exactly opposite.
It's working in the reverse gear. Another question that has come was about the possibilities of nasal or transdermal delivery of this vaccine.
Probably not. They are giving to the, you all know about, they want now to deliver in our lymph nodes directly to get the immediate immunoresponse. So I never come to any company who is producing such vaccines. So all might be intravenous vaccines.
Dr. Thorat, there is a concern of the high efficacy of this vaccine. Moderna claims 94%, Pfizer is claiming 95%. Normally, vaccines do not claim such high efficacies. So, that kind of report, you know, it's like saying 100 upon 100, you start wondering how they got 100 upon 100.
Yeah, so it's
And that
just
is one of the reasons of the public mistrust for this vaccine. Can you
yes,
tell us something about that?
Yeah. So what actually these all vaccines developed within last 9 to 10 months. So for efficacy data, we have to wait for 2-3 years. Vaccine trials actually, it's for very long protocol. They do phase 1 for 1 year, phase 2 to phase 3 is 3 to 4 year trials.
Yeah.
So it's just a 6-month trials. Within 6 months, they found it's efficacy 90%. But we don't know how be the next year or 2 year or 3 year. So we need a time to understand real efficacy. Initially, it might be 90%, but with the time being, it might be 50% as well. Like flu vaccine. Flu vaccine have just 50% efficacy. And every year, this flu virus influenza is mutating. So they are again researching how much mutated, what type of mutation is there, and they are redeveloping new vaccines every year. And every year, they are vaccinating people for flu vaccine. So this will be consequences in future. We don't know the real efficacy of corona vaccines.
Is there any study or any research that suggests that this, because of this mutation of the corona, we may need to kind of upgrade the vaccine more frequently?
Yes. Just 2 days ago, UK Health Minister announced that they found one mutated virus in some hospital. One patient have different coronavirus than the other. So there might be some mutation now started, and more study we need. From the first virus when they detected in China, and now in UK, it's completely different. So it's kind of be like flu vaccine in future. They have to, research every year or every time and make a new vaccine or make a new genetic makeup for vaccines.
Okay, Dr. Thorat, last question before I hand you over to Dr. Gulbaki. There are two sections in society. One are the people who are waiting for this COVID vaccine because they think that will kind of relieve them of all the mask and the kind of limitations and restrictions. And the other are those people who are fearing the vaccine because it has come too good, too much, too fast. What would you say would be your response to both these groups of people?
So fortunately, I have accompanied with these both people. So some of my colleagues, they are eagerly waiting for vaccine. Even they are researchers. And some are opposing, even they are again researchers. So they are saying these vaccines have more consequences, like I said, mRNA vaccine, if it interfere with our genetic material in cells. So this may cause the cancer or some other diseases in future. So some people in Europe, India, not in India, but I would say in the US, they are worried about that as well because they know the scientific process more than us. And some are eagerly, they are saying, okay, whatever may be in the future, what we need right now, only hope is the vaccine. So don't worry about the future and herd immunity and other tricky things. Get vaccinated and get rid from this disease.
True. I think it's all going to be a question of educating everybody and making them aware about what is the situation. Yeah. Thank you so much, Dr. Thorat, for so kindly and patiently answering all these questions. And I'll request Dr. Arvind Gulbaki, who has been, I think, your colleague, and he's our research coordinator, to please deliver the vote of thanks. Dr. Arvind Gulbaki.
Thank you, ma'am. I'm here. Honorable Chancellor and President, Dr. Sanjay D. Patil, sir. Respected Vice Chancellor, Professor Rakesh Mudgal, sir. Pro-Vice Chancellor, Professor Shirpa Sharma, madam. And today's guest speaker, Dr. N.D.Thorat, and Dean Professor R.K. Sharma, sir. Research Director and Dean CIR, Professor Lokhande. Registrar, Dr. B.V. Bhosle, sir. And distinguished faculty members and dear research scholars. Very good evening to all of you. I, Dr. Arvind Gulbaki, Coordinator, Research and Development, on behalf of D.Y. Patil Education Society, deemed to be University Kolhapur, and IQAC, feel honored and privileged to get an opportunity to propose a vote of thanks on this occasion. I, on behalf of DYPU Kolhapur and IQAC, I extend a very hearty vote of thanks to Honorable Chancellor and President of D.Y. Patil Education Society, Kolhapur, for his great support and encouragement. I would like to express my gratitude to our Vice Chancellor, Professor Mudgal sir, for welcome talk. I would like to thank IQAC coordinator and the Pro Vice Chancellor, Professor Shimpa Sharma Madam, for organizing this wonderful webinar, which is well focused on the current problem, and also introducing our today's guest, who is Dr. Thorat. I am very thankful to Dr. Thorat for his wonderful talk on the COVID-19 vaccine development. He gives an in-depth knowledge about the vaccine development and the current status of the many companies. And he also solved the many answers to all the questions, I think, from the audience. We are extremely grateful to the registrar and the finance officer for their support and encouragement. Now, the most important, my special thanks to our IT department, Mr. Suraj and his team for the technical support. I convey thanks to all distinguished delegates and researchers for attending this wonderful webinar. Thanks to all of you. Lastly, I would like to share one important information. The link is available in the chat box. You can just fill the feedback, which is very important for our IQAC department and university to arranging the kind of wonderful seminars, webinars in future also. So please fill the feedback. Thank you once again, all of you. Thank you.
Thank you very much.
Thank you so much, Dr. Gulbaki.